So in this session, we're going to discuss accessory organs of the digestive system. So in the other video, I discussed about accessory organs found in the mouth or associated with the oral cavity, that is salivary glands. We talked about teeth, talked about tongue. But in this particular one, we'll focus about those associated with the small intestines. We're talking about the pancreas, we're talking about the gallbladder, we're talking about the liver. Those are accessory glands that help to finish other functions of the digestive system. So the first one we'll look at is the pancreas. So saying the pancreas is both endocrine and exocrine gland. It is found just posterior to the stomach. It is sometimes referred to as the bed of the stomach. So the stomach lies on the pancreas. Posterior to the pancreas are the great vessels. We have inferior vena cava. We also have aorta. Okay. Right, so we're saying posterior to the pancreas are the great vessels, namely the inferior vena cava and the abdominal branch of the aorta, which is referred to as the abdominal aorta. Then we find that right lateral to the pancreas is the duodenum. So the concavity of the duodenum serves as where to find the head of the pancreas. Then we see that on the left lateral, the tail of the pancreas borders with the spleen. So that being said, let's look at parts of the pancreas. So the pancreas has a head, which is this part. The most wide part is the head. Then this is the body, the midsection. This is the body of the pancreas. Then further there is the tail of the pancreas. Now we also observe that on the head region, a small part of the head coils going posterior to the pancreas. We call that as the uncinate process of the pancreas. So that is the uncinate process of the pancreas. Then we see that through the entire length of the pancreas, from the tail, body, and head, the main pancreatic duct runs through. So this is a main pancreatic duct with small ducts joining it. Then towards the head, the pancreatic duct gives off another duct, which is known as the accessory duct of the pancreas. So the main duct is known as a Weyer's duct. The accessory is known as Santorini's duct. So we have duct of Weyer's and duct of Santorini. It is the main duct or the Weyer's duct that is connected to a common bile duct there at the hepatopancreatic ampulla there. So the two fluid will use one valve or one exit to enter the small intestine, which is the hepatopancreatic ampulla of Vata here. Then the sphincter that closes that ampulla is sphincter of Oddi. That is O-double-D-I, sphincter of Oddi, as seen here. Then this lump in the duodenum where the sphincter of Oddi is found is known as a major duodenal papilla. Then there's a smaller lump above where this accessory duct enters through, known as the minor duodenal papilla. So in short, there are two papillae in the duodenum. The major one is for the main pancreatic duct. The minor one is for the accessory pancreatic duct. All right, so that is it. So we're saying there are two ampullae, the minor one and the major one here, the papillae, sorry. And we said that the major duodenal papilla, that's where the upper gut is divided from the mid gut. So what else are we looking at in the pancreas? So we're saying the pancreas, apart from being an exocrine gland, meaning it's producing its contact into the duct, like the major duct here, the pancreas also has endocrine portion, produces glucagon and insulin as hormones. So here we are seeing now the gallbladder present. So apart from the inferior vena cava and aorta at the back there, we're seeing that the gallbladder is now showing here. Okay, this may not be the actual position of the gallbladder in a living subject, but nevertheless, this is just helping us to understand that these structures are nearby and they're associated with each other. Needless to say, there will be a liver here, just superior to the gallbladder. There's a gallbladder, this is a pancreas. All these are known as accessory organs of digestive system. Okay. So we're saying the exocrine part of the pancreas secretes pancreatic juice, and this juice contains various enzymes. These enzymes finish digestion of carbohydrates, proteins, and fats. Okay. Whereas the endocrine part is denoted by the presence of islets of Langerhans. So on the pancreas, apart from having these acini, okay, these are acini glands. If you look at this, these are simple acini glands. If you know, branched acini glands. If you check the lesson we did under glands, you'll be able to determine how to classify glands. Now, we are seeing that apart from having these exocrine glands, there is also these islands of cells. These cells are endocrine cells. So they are beta cells and alpha cells. Beta cells are staining basophilically here, hence called beta. Then the orange ones are staining acidophilically, hence called alpha cells. Beta cells produce insulin, alpha cells produce glucagon. Now, those chemicals are not being transported by this duct. Those chemicals are transported by capillaries found within these cells. So we can see those red capillaries there. Even here, there are some capillaries. So that's a capillary, another one here, another one there. So blood vessels acting as ducts here. So in terms of content of pancreatic juice, it was covered under small intestine. So I would refer you to that presentation. Otherwise, the juice from the pancreas helps to finish digestion of all these macromolecules, carbohydrates, proteins, and fat. What about the liver? So liver, we're saying, is the largest organ or gland inside the body, weighing about 2.5 or 2.3 kilograms. Okay. So that is a liver with two major lobes. Then it has two more minor lobes. So the major lobes are divided as the left and right lobe. So this is the left, this is the right lobe, sorry. Then this is the left lobe, divided by the falciform ligament. This is a falciform ligament here. Then that is right lobe, left lobe. Now we're saying the undersurface of the falciform ligament, this ligament separating left from right, is a ligament that was previously serving as umbilical vein. So this is umbilical vein and its remnant. In a fetus, it is a vein and it is channeling oxygenated blood from the placenta to the inferior vena cava. Now, after birth, that vessel is no longer functional because the umbilical cord is cut from the placenta. So the vein undergoes atrophy and then remains as ductus ligamentum. Okay. Now, after that, this ligament is seen in the inferior surface of the falciform ligament. Okay. Then the other two small lobes, they are seen on the undersurface of the liver. So we have the caudate lobe and the quadrate lobe. So if you're inspecting the liver on its undersurface, superior, the caudate lobe will be superior, then inferior to it will be the quadrate lobe. Now the quadrate lobe is next to the fundus of the gallbladder, this part here. Just next to it is a quadrate lobe. Superior to the quadrate lobe is a caudate lobe here, which is seen up there. So these are small lobes, but the major ones we concern ourselves are the left and right lobe. What is the function of the liver? Okay, so apart from that, we see that there are structures entering the liver. We have the portal vein, we have the hepatic artery, and also the bile duct. So these three structures enter in the liver through the hilum. Here, that is the hilum of the liver. And collectively, they are known as portal triad. Triad meaning three. Hepatic artery, portal vein, and gallbladder. Now, hepatic artery is carrying oxygenated blood from the heart into the liver, whereas portal vein is carrying deoxygenated blood from the digestive system. So blood running through the portal vein is rich in nutrients from the digestive system. After absorption, those nutrients will go to the liver via portal vein. But the two blood will mix. Once these vessels enter in the liver, hepatic artery and portal vein, they mix to form hepatic sinusoids. These are special capillaries found in the liver. So hepatic sinusoids, as we'll see much later on, will carry both deoxygenated blood and oxygenated blood from those two sources I've mentioned. Deoxygenated blood is coming from the digestive system, but it has high nutrients. Oxygenated blood is coming from the heart, but with low nutrient levels. Then they mix in the liver lobules, and then metabolism occurs from there. Okay. So let's look at liver lobules. So the liver is divided in two lobules. So when you cut the liver and examine it, we find that the liver has lobules. Now, these lobules are said to be hexagonal in shape. The lobules are said to be hexagonal in shape, more like six-sided corners. 1, 2, 3, 4, 5, 6. Another corner which is not shown here. So this is one lobule. Here there will be another lobule here, another lobule. So this is a sample of the liver gotten from a pig. Here is a sample of the liver gotten from a human being. Nevertheless, the structure is similar, except that in the liver of the pig here, we see that lobules are much clearer to be seen because you can see connective tissue there separating one lobe from the other. But in the human liver, the connective tissue that separates one lobule from the other is not so dense, and therefore it's difficult to see the corners of one lobule. And therefore, we just use the central vein to identify a liver lobule. So in the middle of each lobule, there will be a central vein. You see that? That is central vein here. So that's what we use to identify liver lobules. More clearly shown here. So here we have 4 liver lobules. 1, 2, 3, 4. Each with 6 sides. Then we are saying in the middle of each lobule is a central vein. So that's central vein, central vein, central vein, central vein. Now, we are saying 3 vessels entered in the liver. That is hepatic artery, portal vein, and bile duct. By the way, the bile duct was leaving the liver. Now, what is happening here is we call them as a triad. This is a portal triad. Hepatic artery, portal vein, and bile duct. So the 2 vessels, artery and vein, they are taking blood into the liver lobules. So they keep on dividing. So each liver lobule is guarded by portal triad. So at each corner here, there will be a portal triad, portal triad, portal triad, portal triad, as seen here. Central vein surrounded with portal triads. 1, 2, 3, 4, 5. The 6th one was supposed to be here. Now, what happens is blood in the hepatic artery and blood in the portal vein mixes here to form a sinusoid. You can see this is oxygenated blood, that is deoxygenated blood. There is a sinusoid. It is a sinusoid that connect to the central vein. So by the time blood reaches into the central vein, these cells here, the hepatocytes, would have used that blood. So this is the same structure now, just magnified. Artery and vein, each one going, and then they meet there. The moment they meet, what you have is a sinusoid. This is a special type of capillary. We call them discontinuous capillaries because they have large gaps which allow migration of nutrients from the digestive system to enter into the hepatic tissue so that metabolism can take place. So what we have here is mixture of blood there. Now, within the sinusoids, we find special type of macrophages. You can see those in brown. Those are macrophages. Here in the liver, they are called Kupffer cells. So all sinusoids in the liver have macrophages, and macrophages are acting there as guards. You know, they are trying to destroy any old red blood cells are removed by these macrophages called Kupffer cells. So by the time blood reaches the central vein, it is cleaned by the macrophages. Next, these cells, the hepatocytes, would have absorbed the nutrients, metabolized, and given new product to this blood, then taken to the inferior vena cava. So all the central veins, this 1, 2, 3, 4, will finally make one vein, what we call the hepatic vein. It is a hepatic vein that is now connected to the inferior vena cava, and that blood is taken to the heart. So this is the structure of the liver lobule, which you must be acquainted with. These are hepatocytes, and you see that hepatocytes are in columns. They are like that, in columns. Then in between hepatocytes, we find the sinusoids. So that's the sinusoid, 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 which carries a mixture of blood. Because of that, liver lobules have been classified as following. So we have what we call a classic liver lobule. So a classic liver lobule simply represents the flow of blood in the liver. So blood is coming from these two vessels, runs through the sinusoids to reach the central vein. Then we have a portal lobule. A portal lobule is concerned with the flow of bile. So bile moves in the opposite direction. Blood was going towards the central vein. Bile is being formed by these cells closer to the central vein, even cells in the middle here and cells there. So instead of going this side, since there is no duct to collect bile, bile moves in the opposite direction. Then finally enters into the canaliculi. So these green tubes we are seeing here, or ducts, we call them bile canaliculi. When they form a large duct, we call it now a bile duct, which is part of the portal triad, these three structures there. So a liver lobule is concerned with the direction or flow of bile, which is in opposite direction to blood. The last one is hepatic acinus. Hepatic acinus or acina is concerned with concentration of oxygen in the liver. So we are saying blood in the portal artery, in the hepatic artery is oxygenated. Blood in the portal vein is deoxygenated. So, and the portal triad are found away from the central vein. This is central vein, portal triad is here, there, there, there. So what it means is cells that are closer to the hepatic artery, these cells here, all that area, will receive the highest concentration of oxygen. Cells in the middle will receive minimal amount of oxygen. Cells that are closer to the central vein, because they are now far away from the artery, receive the least concentration of oxygen. So you find that most of these cells closer to the central vein are performing anaerobic respiration, whereas those in the middle and those closer to the portal artery, hepatic artery, are performing aerobic respiration because they have highest concentration of oxygen. So I'm repeating that again. Hepatic acids deals with concentration of oxygen in relation to the portal artery, because it's the one that is bringing oxygenated blood. Therefore, cells that are in the periphery of a liver lobule receive the highest concentration of oxygen. Cells that are closer to the center of a liver lobule receive the least amount of oxygen. Then portal lobule, we're looking at the flow of bile. So bile is moving away from the liver lobule to go into the portal, the bile duct, leave the bile duct and go into the gallbladder. So the cells producing bile are the hepatocytes. Then the gallbladder will just store it. Then classic hepatic lobule, you're looking at the direction of blood. So it is the portal artery and the hepatic artery and portal vein that bring blood to the liver lobules, and blood will move towards the center of the liver lobule to go into the central vein. So that's all about the liver. Okay. Then if you look at the bile canaliculi, I was saying these are hepatocytes, by the way. So these cells are the ones producing bile. Then these small ducts are the bile canaliculi, which form the bile ductule. The bile ductules merge to form the bile duct, which leaves the liver to go into the gallbladder. So we're going to have left and right bile duct. The left coming from the left lobe of the liver, the right coming from the right lobe of the liver. Okay. So this is still a liver lobule. Function of the liver is quite many. The liver has so many functions. Among them is metabolism of all macromolecules, that is carbohydrate, fat, and proteins. Next is breakdown of old red blood cells. I mentioned Kupffer cells, those that are found in sinusoids. They help to remove all the old red blood cells. Next is detoxification. The liver is the main organ for detoxification. Then inactivation of hormones is the liver, synthesis of vitamin A, and storage of substances. For example, all fat-soluble vitamins are stored in the liver. In addition, there's iron, copper, some other water-soluble vitamins like riboflavin, niacin, vitamin B12 can be stored in the liver. Production of heat. So as the liver is performing metabolism, the byproduct is heat, and that helps to warm up the body. Next is synthesis of bile. Bile is produced by hepatocytes, then it is stored in the gallbladder. So here we have this illustration showing us how red blood cells are destroyed. So red blood cells are destroyed by the liver and the spleen. So what happens is when the red blood cells are destroyed, hemoglobin is broken down into heme and globin. Heme is further broken down into iron and bilirubin. Bilirubin has to be conjugated in the liver. That's when it can now be excreted in feces as stechobilinogen and in urine as urobilinogen. So you see that the color of stool and urine is influenced much by bilirubin, which is a byproduct of red blood cell destruction. So if there's interference with conjugation in the liver or transportation of bilirubin from the liver into the intestine, or transportation of bilirubin from blood into the kidneys, finally into urine, there will be accumulation of bilirubin in blood. And what will happen next is bilirubin will be deposited into tissues, causing a condition known as jaundice. That occurs in patients having malaria, for example, when red blood cells are being destroyed by the malaria parasites. It can also occur in premature babies where they have insufficient amount of enzymes to conjugate bilirubin. So unconjugated bilirubin leaves the blood vessels and stains tissues, condition known as jaundice, which has the potential to damage the brain of a neonate. Okay. So what is important again is just look at site of anastomosis. So this one, I think we'll discuss it much more when we have the revision course under a live session. Let me quickly touch on the gallbladder. So saying the gallbladder is pear-shaped and is attached to the inferior surface of the liver, posterior and inferior surface of the liver. So this is a gallbladder having the following parts: fundus, body, and neck. Then in terms of that, we have a duct connecting to the gallbladder known as cystic duct. These are the two bile ducts leaving the liver, left and right. Then we have a hepatic duct when the two meet. Then cystic duct transports bile to the gallbladder and back into now the common bile duct. It's a common bile duct that transports bile to the digestive system. Okay. So in terms of histological features, we say the inner lining of the gallbladder is highly folded there, more like the stomach. So it has rugae as well. It has these folds, like you can see. These are folds. Then similar to the general structure of the digestive system, where you have mucosa, submucosa, and muscle layer. Then finally adventitia. Now, when it comes to the gallbladder has a very thin submucosa, and some books actually say that it is absent. So it's easy to identify this as not being the stomach because it has no submucosa. As you can see, that is just mucosa and lamina propria. Next is muscle. After that is adventitia. So that is the gallbladder. Okay. So we're saying the biliary system now is this system connecting the liver, gallbladder, and intestines. So this is a biliary system. Liver above, we have gallbladder here, pancreas, and small intestine. So this is connected by tubes. Here we have pancreatic duct. There we have cystic duct. Here we have hepatic duct from the liver. Then they meet in the common bile duct. Common bile duct, it will meet with pancreatic duct here at the hepatopancreatic ampulla of Vata. Then finally enter the small intestine. So the whole lot here is a biliary system, okay, which is a subdivision or a subsystem of the digestive system. Okay. So function of bile, we're saying function of gallbladder, storage of bile, then concentrates bile and releases bile when there's fatty meal in the stomach. And we know that bile simply emulsifies fat, so that now, pancreatic lipase can act on the fat, so that now complete digestion of the fat is achieved. Otherwise, this is where we end with accessory organs of the digestive system. The next topic we'll look at now, we'll look at the respiratory system. So this is all for now. So if there are any questions, even from previous discussions that we had, just feel free. Here, you can ask questions in the group, or you can ask questions directly in my inbox. We'll be able to respond to that.